The center of a neuron is the cell body along with the nucleus inside. The extension of these cell bodies are dendrites, which are connected to other neurons and taking information in the form of neurotransmitters. This description of the center of the neuron is to receive information. The center then transitions to the axon, where it transmits the received information. This begins at the axon hillock and makes its way to move through the axon terminals. In the terminals, there is a synapse that connects us to the other neurons, and the signals would move from the starting dendrites in the cell body, and then the information is transmitted to the next neuron through a process called action potential. In order for information to travel quickly, the axon has wraps of myelin sheets, which is a Schwann cell that's wrapped around. This occurs by putting a voltage-gated channel in between the sheets that is called the node of Ravenier, and it acts as an insulate to the nerve. This allows the info to travel more readily down the axon. The way we take in information is there will be another neuron that is connected to both the cell body and the dendrites of the cell. There will be a gap between these two, which is called a synapse, which simply means a connection between neurons. Action potential. All the summation of neurotransmitters is either going to fire an action potential down the axon or not. In resting potential, there is a separation of charges across a membrane on the surface of the neuron and can measure the voltage. At this particular point in action potential, ions of different charges are jumbled up with one another. A leak channel will be a gateway to let a dominant charge seep out, as well as a chemical gradient from the inside to the outside. Say if you focus on positive ions inside the membrane, the chemical gradient would decrease as the positive ions flow outside. This allows an electrical gradient to form as more positive ions leave. Ultimately, an electrochemical gradient could form because not all ions will be in the same part of charge. The ions inside became ultimately positive through polarization because of the mass amount of negative ions. In conclusion to resting potential, negative 70 millivolts is established because we have a separation of these ions on the inside and outside, and there are different electrochemical gradients are established. That difference in the charge and therefore have a resting potential. In order for action potential to occur is to depolarize the resting neuron. This means to cause a big enough change in its membrane potential that it'll trigger the voltage-gated channels to open. It starts with the neuron sitting at resting state with all ion channels closed and the inner voltage resting at negative 70 millivolts. When that happens, there is some type of environmental stimulus that occurs, which triggers these sodium channels to open, increasing the charge inside the membrane. The stimulus and resulting change have been strong enough to cross a threshold for true action potential to kick in, and the threshold will be negative 55 millivolts. At that threshold, the voltage-gated sodium channels open, so all of the positive sodium ions rush in, making the cells massively depolarize, so much so that it ends up being 40 millivolts. Action potential triggers a biological chain reaction which sends that electrical signal down the axon. Synapse is the meeting point between two neurons. When action potential occurs, it sends an electrical message to the end of the axon. That message hits a synapse that then translates or converts it into a different type of signal and flings it over to another neuron. These synapses are able to change and adapt in response to neuron firing patterns. Some synapses are electrical, which mean immediate, and others are chemical, which are controlled and take longer. Electrical synapses send an ion current flowing directly from the cytoplasm of one nerve cell to another through small windows called gap junctions. In chemical synapses, neurotransmitters are used, or chemical signals is that diffuse across the synaptic gap to deliver their message and effectively convert signal in steps from electrical to chemical back over to electric, which allows different ways to control that impulse.